All right, welcome back, you guys. Today we're going to finish up chapter 12. Remember, chapter 12 is on gene mutations. Now, to recap, we talked about how mutations are changes to the actual DNA, the bases, whether it's the order of the bases, or we can delete things or add more. But it's a change to the original DNA structure, to the bases. And if it's in a part of the DNA that codes for proteins, it can have some pretty big effects. We mentioned a few of them, like sickle cell disease and beta thalassemia, which are when there's a mutation in the gene that produces the beta globin subunit protein, which is part of hemoglobin. We also talked about collagen disorders such as L-Port and Marfan syndromes. Now, collagen is that flexible but strong connective tissue, and what also allows, or in connective tissue, sorry. It's also a protein found in structures such as organs and your blood vessels to allow for that flexibility to move, but also to stay strong. We've also talked about types of classifications of mutations. We have substitutions, also known as point mutations, where you switch one base for another. And we talked about the different varieties of point mutations. We've also talked about frame shift mutations, which is when you have an insertion or deletion and it causes all the bases beyond that point to shift over, to move. And that can actually change all of the possible amino acids beyond that point. We did talk about a few more, like transposons and pseudogenes and all that. But mostly I just want to start talking about section 6 and 7. So 6, as you can see, is on the importance of position, and 7 is on DNA repair. So let's get going, shall we? All right, there it is. So the exact location within a gene that a mutation occurs can lead to different effects. So depending on where in the gene, not just what happened, not just is it a substitution or a deletion or an insertion, but also where in the gene. Is it closer to the front of the gene? Is it in the middle? Is it closer to the end? You can actually see differences that can arise from that. We have some examples. Some of the interesting ones are different globin variants. Again, globin as in hemoglobin. So there are different variants that can occur in either the alpha or the beta subunits. And one of the most common symptoms is that it can cause anemia, which means that it can't always carry as much iron. And this can also affect how much oxygen can bind to it, or it can be a silent mutation. For instance, there is 2 forms of a particular mutation, and if you have one form of it, HBS, it leads to sickle cell disease. But if you have a slightly different form of it, still a change from the original DNA, HBC, you don't end up with sickle cell, but the other, remember we said sickle cell disease, something that they've noticed is that people with sickle cell disease tend to be resistant and immune to malaria. But what they notice is with this particular variant in HBC, what happens is the individuals are immune from malaria still, but they don't have the sickle shape. It's still slightly changed, the blood cells are still slightly changed in shape, but not to the same degree that it causes the problems that it does in sickle cell. However, because of that slight change in shape, there is still the protection against malaria. There's also something called a methemoglobin gene, and that helps with how the blood underneath the skin is kind of complex, so I apologize, and I'm trying not to give like this really long spiel on it. But what happens is it actually affects the pigmentation of certain parts of the body, like the skin or inside the mouth, because of a mutation within the actual methemoglobin, which is related to hemoglobin. One example is black mouth of Japan. So one variant of this causes a darker pigment on the inside of the mouth, so like the lips, the teeth, the gums, the tongue. It all ends up being of much darker pigment, almost black. Another interesting one, which there's a lot more information on this one, is the blue people of Kentucky. This is also known as cyanosis, and we have a picture right here. So you can see the guy there, the older gentleman. He has this very, very pronounced blue tinge to his skin, and it's because of this mutation in the methemoglobin gene. It causes his skin to appear that blue shade. And next to him is his wife. I believe it's his wife. But you can definitely see the big difference between their skin appearance. So that's what cyanosis is. We actually do have a video on the blue people of Kentucky and the additional resources. So definitely check that out. It's pretty interesting. All right, let's move on. So there's also something called prion disorders or prion diseases. Now, prions are when proteins get misfolded and they become infectious. Now, these infectious proteins, one of the major effects of them is that they can cause different forms of brain degeneration. So they affect the mental capacities and a lot of neurological effects as well. These can be either inherited, so you can have a mutation that caused it caused your body to make these incorrectly folded proteins automatically, or it can be acquired. So a while back, even when I was younger, there was the mad cow scare. So mad cow disease is a prion disorder. And what happens is if you ingest some of these misfolded infectious proteins, these prions, then they can actually be absorbed into your system. And they work very similar to how viruses and bacteria work, like how a virus can hijack your cell to make more of the virus. But what these prions do is they hijack your cells to make more prions. So they force your cells to make more of these infectious proteins, and they don't work properly. And that's what causes a lot of the neurodegeneration. So certain mutations in prion gene predisposed individuals to prion. Can you tell it's a Friday? Certain mutations in prion gene predisposes individuals to prion disorder. So what that means is that there's a mutation that makes it more likely for these particular misfoldings of proteins to occur. Now, doesn't necessarily mean someone with this particular mutation will make prions. No, but it means that they are much more likely to misfold the proteins, causing those particular proteins to become prions, and then that will lead to a prion disorder. So we have some examples here. Fatal familial insomnia is an example. Crutchfield-Jacob syndrome, Kuru, we mentioned mad cow disease. What they have noticed though is that mutations that occur more so at the end of the prion gene or in the third codon position. So remember, a codon is 3 bases in the mRNA. And if it occurs in the third base, or if the mutation occurs at the end of the gene, then it really doesn't alter the protein, not in such a way that it'll become a prion and become infectious. So like we said, it's the position that matters. If this mutation occurs earlier in the gene, or it's in one of the first 2 bases of the codon, then you're more likely to develop a prion disorder. But if it occurs at the end of the gene, it's a lot less likely to develop into the prion disorder. All right, and we also have these things called conditional mutations. So when we talk about things being conditional, we mean in terms of, that there are certain situations or circumstances that need to occur in order for the effect to be seen. And that's the same with a conditional mutation. These will only express themselves. So they are mutations that will only express themselves in certain environments. And that's because there's an environmental trigger for them. So the mutation is already in the DNA, it's in the genes, it's in the DNA, but it's not being expressed because it only gets expressed when the organism is exposed to certain things. So some examples are, there's the pollen of a particular plant in Baghdad, fava beans, and there are also certain anti-malarial drugs. Now, what happens with these is, specifically, it's in the G6PD enzyme. And there's a mutation in the gene for that enzyme. Now, here's the thing. If you're not exposed to any of those, then the mutation does not get affected. It's just going to keep doing what it does normally. But in the presence of one of these, either this particular pollen or fava beans or certain anti-malarial medications, then what they notice, sorry, the sun's getting in my eye, is that it causes this mutation to activate. And what happens is hemolytic anemia. Now, this is very dangerous because it can actually cause the blood cells to burst. They can actually start bursting and dying. So it is a very life-threatening, potentially fatal outcome. And it's just the body reacting. Kind of a bad analogy would be like an allergy. If you don't go outside, you don't get exposed to the pollen, you don't produce the antihistamine or the histidines, and then you don't, get all sneezy and whatnot. Well, most of the time, someone, let's say potentially if I have this mutation, I may not even know it because it's not going to be expressed. I don't live in Baghdad, so I'm not exposed to pollen from this particular plant. I don't eat fava beans. And as I live in Maryland, I don't really need to take any anti-malarial drugs. So since I'm not really exposed to any of these conditions, I'm not going to see these potentially harmful effects of the mutation because there's nothing to trigger it and cause it to be expressed. That doesn't mean all conditional mutations are horrible and scary and whatnot. Look at those cute little bunnies right there. So there are bunnies, and actually some cats, like Siamese cats, where they have the lighter fur and then they have darker portions like around the ears, the face, or the paws, like we see in the bunnies here, and also with Siamese cats. Now, here's how that actually works. There's a particular protein, a pigment that is created by the body that gives those areas the darker coloration. However, it's also affected by the environmental conditions with which these bunnies are born into. When it's colder, then, trying to remember, bear with me. Like I keep telling my kids, I'm not an expert in everything. So I believe it's when it's colder, it causes more of that enzyme or that protein, that pigment to be created. And that leads to the much, much, much darker, almost black ears, face, and paws. If it's warmer, not as much of it is created. And that means that it's going to be a lighter grayish color as opposed to the darker, almost black color. So the conditions in which those bunnies are born, if it's colder, they'll have the darker sections. If it's warmer, then there'll be more of a lighter color, like more of a grayish. So again, not all conditional mutations are bad. Look at those cute little bunnies. It's just that certain environmental conditions need to be met in order for those traits to be expressed. Yeah, right. Now let's move on to section 7, DNA repair. We've mentioned previously that our DNA does have some repair mechanisms in order to help fix it if there are mutations that occur. So, DNA repair. Many genes encode enzymes that search for errors in DNA and corrects them. Woohoo! We can actually fix a lot of things, which is why, for the most part, just in terms of the amount of times we replicate our DNA or we step outside, it's amazing we don't have more mutations or things like that. But the reason why we don't is because of these repair mechanisms. So here's an example. UV radiation causes dimers and pyrimidines, creating an extra covalent bond and kinking the DNA, which disrupts replication. So what that means is when you go and spend too much time out in the sun gives off UV light. Now, if you look at this picture here, the UV light is actually going to cause these thymines, remember thymines are pyrimidines, to break their bonds with the adenines and form covalent bonds between each other. Since they're bonded here and no longer with the adenines on the other strand of DNA, it causes the the sugar phosphate backbone to kind of bulge. It kind of like, instead of being nice and flat, it's going to bulge up and bubble out. Well, when it does that during DNA replication, the proteins like DNA polymerase can't effectively move along to read and replicate the DNA. So it affects how DNA is replicated. But what they're saying is that we have enzymes that search for these mutations. For instance, there are particular enzymes that search for these dimers, break the bonds between the thymines, and allows them to rebond with their complementary adenines on the other strand. So we'll go back to being a normal DNA. So what are some correctional techniques? Well, this first one we actually don't have, sadly. So photoreactivation. There is a particular enzyme called photolyase. And what it does is it actually absorbs energy from light, like the sunlight, and breaks the extra bonds. So this is a particular way that other organisms like plants can fix those thymers. We don't have this particular method. We have other ways to fix it, but this is one way that could work. And that kind of makes sense that plants would have something like this. They need to be out in the light all the time. They have to do photosynthesis. Me, I can stay inside like I am now and just kind of chillax. But plants, they need sunlight in order to live, to do photosynthesis so they themselves do not die. So as they are exposed to UV light pretty frequently, they have these particular enzymes, the photolyases, and they absorb energy from the light. So they're using what caused the mutation in the first place. They're using the UV light to gain energy in order to break the bonds that are caused, to break the dimer bonds that were caused by the UV light. So that's just kind of cool to me. It uses energy from light to fix the mutation that was caused by the light. All right, so we don't have that. Plants do, lucky them. We do have these next couple. So mismatch repair. There are certain enzymes that will actually kind of go through the DNA. It's going to look for places where the sugar phosphate bulges out, or sorry, I keep hearing something. So where it bulges out or it kind of loops or it's kinked, it's going to look for areas where the sugar phosphate backbone is not nice and straight. And specifically, it's going to look for it in the newly replicated DNA. So this is after DNA replication has occurred. These enzymes are going to go and search out along the sugar phosphate backbone to make sure everything is correct. Now, if there's a bulge or anything like that, that will indicate usually that there's some kind of mispairing. So we know adenine and thymine should be bonded together, but if it's adenine and cytosine or adenine and guanine, then it won't really pair up as well, and that's what could cause those bulges. So they're going to search that out in order to cut out the wrong base and put in the correct one. We also have what's called excision repair. There's two types of these, and this will actually cut out sections of the DNA and repair it in sections. So instead of just looking at like one or two mismatched bases, it's going to actually look at sections that need to be replaced. So let's talk about a couple examples of that. So nucleotide excision repair is you have a bunch of enzymes that work together to repair sections that are, like 30 nucleotides long, give or take. So 30 nucleotides or more. It's going to look at these bigger sections, not like super big. I mean, if there's a major section of DNA that is messed up, the enzymes just might scrap that whole thing and try again. But in this case, if it's a somewhat large section, like 30 bases or more, give or take, it's not going to be like thousands of bases long. But what it will do is actually these enzymes will come, they'll cut out that section, they will remove it, and then they will go and replace it. Then we have base excision repair, which is when there's just more than one, but a few bases that need to be replaced. So 5 or less nucleotides. And again, the enzymes work together to try to repair that, kind of like down here. There's these 2 nucleotides that are not correct. The enzymes come in, cut them out, and then the correct bases will be put in. Well, what happens if the repair goes wrong? So there are some diseases and disorders that occur because the repair mechanisms didn't do their job properly. So let's take a look at those. Now we're actually going to talk about specific examples of disorders. These are some examples of excision repair disorders. So the first one is xeroderma pigmentosa, or XP. That's this child up here on the top right. So what happens is it causes blisters and skin cancer from sunlight. And the reason for this is because of a deficient excision repair. So what happens is you go out in the sunlight, even if you spend a couple hours, you might be developing some of those dimer mutations, but your cells and your enzymes naturally go through and repair them. And what if you can't repair them ever? That's essentially what xeroderma pigmentosa is. You end up with these blisters because you're constantly getting these dimers and you can't fix them. And then the constant buildup of these mutations caused by the UV rays then lead to skin cancer. We have trichothiodystrophy. Now, this is an interesting case because an individual with this will actually start with normal development, but then as they start getting, or once they hit a certain age, then their appearance suddenly starts to look more aged. So it could be the individual's 10 years old, but maybe they look 20. And I'm not saying that they necessarily look like a full-grown adult, but this individual at the bottom in box A, because of the texture and tone of the skin, the wrinkles, we might say he might be like, in his 50s. That individual might only be 20. It's that aged appearance. Sometimes, or a lot of times, there's also severe effects on mental development, which could lead to mental retardation in terms of a lot of times when we develop mentally, we gain more cognitive skills as we age. So we're able to first understand that there's more than just us. Babies are all about me, me, me. But then as you age and as you grow up, your brain develops. You start being aware of others. You start developing critical thinking, comprehension. But what if you never develop that past the age of 7? And that's just what we mean by that. Essentially, mental development stops at a certain point. They can also cause dwarfism in some individuals. It's not every individual will have it, but there are cases where dwarfism does accompany some variations of trichothea dystrophy. Now, what causes this is a faulty nucleotide excision repair, sometimes in conjunction with a faulty base excision repair, sometimes not. So if it's just a faulty nucleotide excision repair, then you'll get one variation of this with particular symptoms. And if it's the variation where you have both faulty nucleotide and base excision repair, then you get another variation of it. Oh, that's right, I moved my camera. Down there. All right. So mismatch repair disorder. So there's something called hereditary non-polyposis colon cancer, or HNPCC. It usually affects 1 out of 200 people, and it is a deficient mismatch repair. So what happens is the enzymes, as they're checking the newly developed DNA, they may not notice a bulge in the DNA, or they might notice it but can't fix it. Well, that affects how the DNA gets replicated next time, and it can lead to, as you see here, these tumor growths within the colon. And these growths are going to affect depending on when it gets found. If it's found earlier on, they can possibly cut it out and be fine. But again, because it's in the DNA, and it's because of the DNA not being able to make the proper enzymes, or those enzymes are unable to fix the DNA, they are likely to come back. And we also have cell cycle repair disorder. So after mitosis, we mentioned that there are different enzymes that check the cell over to make sure everything went fine. There's an entire restriction or checkpoint at the end of mitosis in the beginning of G1. And if those different checkpoints and mechanisms don't detect anything wrong, or they don't detect, they detect, but they don't actually fix anything, then it can lead to some of these cell cycle repair disorders like ataxia telangiectasis. I'm probably saying that wrong. I've never heard this name. I've only ever seen it written down. So, I'm doing the best I can. So I don't blame my kids for mispronunciation. So AT, and this is recessive disorder. It's because of a defect in cell cycle repair. Now, because it is a cell cycle repair, it does tend to lead to high rates of cancer, because a lot of times after the cell goes through division and goes through mitosis, it checks to see, okay, is everything okay? Everything is not okay, so we'll go to G0, which you see here, and it'll try and fix itself. If it can fix itself, it goes back to G1. If it can't, it goes to apoptosis, which is programmed cell death. Well, what happens with cancer is that the cells don't stop to check themselves. So this is a particular disorder where the cell, maybe it's not that it can't check itself, but it can't fix itself. So instead, it's not quite the uncontrolled cell growth that cancer is, but because of the lack of checks in it, it can lead to very high likelihoods of different types of cancer. It can also affect the balance and coordination because of how it can affect the brain cells and nerve cells. And there also do tend to be red marks on the face and in the eyes, which you do see a little bit here on this picture. All right, so heterozygous. So it is a recessive disorder. However, what they found is that if someone is heterozygous for this, they do tend to have a high risk of certain cancers like breast cancer. So they would have a high genetic predisposition, and doesn't necessarily mean that they will develop these cancers, but they have a higher likelihood of it. And again, because in a heterozygote, it could be that the cell can check itself and it can fix it, but maybe it can't fix all the errors. It fixes more of them, but it doesn't fix them all. So this can still lead to some problems with cell division and cell growth, leading to certain types of cancers. Faulty P53. So first, if we're going to talk about P53 and how it affects us when it's faulty, let's talk about what it does when it's not faulty. It is a protein that can slow down the cell cycle and permit damaged cells to repair their DNA. So remember, we said that there are ways that the cell can actually check itself to make sure everything went fine. So during those checks, P53 is going to be created and is going to check the cell, check the DNA, make sure everything is going smoothly. If everything is not going smoothly, it will actually pause the cell in whatever stage it's in. Usually kind of leaves to the gene knot, but it can potentially check it at other checkpoints. So P53, if these proteins find out that something is wrong in the DNA or the cell, it can actually slow down the entire cell cycle in order to allow those DNA repair mechanisms to do their job. If it cannot fix the DNA, if it's too damaged or there's too many errors in it, then P53 will actually be the protein that induces apoptosis. That's programmed cell death. The reason why it does this is because if there's a lot of errors in the DNA, it's the body's way, it's nature's way of trying to take that out so that it doesn't make you sick. However, it is not a foolproof plan, or nothing in nature is foolproof. Nature is pretty amazing, but it's not a perfect system. So if P53 is faulty, then you can actually have different kinds of cancers arise because there's a particular gene called the tumor suppressing gene. And if there's a mutation in that and it cannot be fixed, the P53 doesn't recognize that there's an error, so it doesn't slow down the cell division, which means that error doesn't get fixed, then it can cause it to just keep getting passed on, and we'll get more and more cells with those errors, and that can lead to tumors and or cancer. So we can actually end up with that uncontrolled cell growth, especially if it's in the tumor suppressing gene. All right, and that is all of chapter 12. I'm going to talk about some additional resources. So we have some of the Science in Pajama videos that I do. There's one that's just generic or very general about mutations. But then we also talk about point mutations in more detail, like the different variety, missense versus nonsense versus silent, transition versus transversion point mutations. And then we also have, so there's a generic mutation one, point mutation one. And then we also have one on frame shift mutations. So if you definitely weren't quite sure what was going on when we discussed frame shift and how that would work, I would check this one out. I would definitely suggest you check this out. I kind of break it down and use different ways to kind of visually show the effects. All right. And then we have some other videos. We have the Amoeba Sisters. They're great. I know it's kind of silly and whatnot, but they do a great job of visually showing as well as breaking down the information. So it's very much understandable. Khan Academy, they have a lot of good information on them too about the different types of mutations. Let's see. Here's the art or the video I was talking to you about the blue people of Kentucky. Talks a little bit about their history. And then we have another Khan Academy, this one on DNA repair. So that could help you understand what those are in a little more detail. But in the meantime, if you have any questions, hit me up in our Google Classroom or through email. You guys know I'm here for you. If you have questions, just let me know. And I will also post the link for this Prezi in the description so that you can access those additional resources should you choose to. All right, you guys. So you take care of yourselves, be awesome, be amazing, and just be the great kids I know you are. All right, you guys. Take care, stay healthy, stay safe, and until next time, goodbye.